Let us talk about structural details of biopolymers further today. So, as we were discussing, there are hundreds of distinct kind of atoms present in any living being, where mostly just six: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. These are the elements that are there. Other atoms such as sodium and chlorine are present, but they are in smaller amounts. So a subtle change in the amount of these ions control certain activities in our body of any living being. So of these small molecules made by joining these atoms, the most important ones are water, obviously, and some inorganic phosphates. So we talked about these phosphates, remember, when we discussed about DNAs, the 4 bases of DNAs. There we saw these phosphates. So the ring structure of these molecules of DNA got a fixed rigid shape. In particular, the bases were flat in structure. Joining the bases to a simple sugar like ribose or deoxyribose, we have seen that how the nucleotides were made. So these are the smaller molecules that we dealt with. There are two more classes of small molecules that have a special interest to us. The first of these will be amino acids, and they are the group of about 20 building blocks based on which all proteins are constructed. So we'll talk about amino acids, amino acid residues, and the bonds involved in these structures today in further detail as we go on making these molecules larger and larger. So in the medium sized molecules present inside any living being, we have RNA, DNA. There are also lipids and polysaccharides. We'll talk about them in the coming discussions maybe. Then after these polynucleotides, where you have RNA or DNAs, we have larger structures than that as well. And these larger structures, the primary of them is obviously proteins. So we have learned the genetic message that DNA encodes and how the polypeptide structure, the genetic code captures the fundamental information flow through heredity. Today we will see that how proteins in their hierarchical complicated structure like primary structure, secondary structure, tertiary structure, how they create and develop different kind of interesting structures and how they maintain the functionalities of different activities that go on inside any living being. So proteins comprise of a largest percentage of organic molecules in our body. Today we will talk about proteins and their building blocks, which are amino acids. So there are they are basically nitrogen containing macromolecules, and in 20 in number, and mostly the amino acids are, and then they are made in form of chains and then folded in complicated structures, the one that you are seeing here right now, like these complicated structures to function in particular ways. So for example, structure of the proteins, they are very important to perform a certain activity. There are some name of some proteins that you maybe already know. For example, collagen and keratin and hemoglobin. These are the common names that we all know, right? So let us start talking about the fundamental unit of these proteins, and those fundamental units are going to be amino acids. So the general structure of the amino acid are depicted in this figure. This central portion is basically called a C alpha carbon atom, and that is attached to 4 groups. This is a carboxyl group, that is COH. This is amino group, NH2, then a hydrogen atom, and finally this is the functional group that makes this a specific kind of amino acid. So these are changes, and accordingly you get different amino acids. So these are attached in a tetrahedral structure across the central carbon C alpha. And most amino acids are also chiral molecules. You know about chirality, so I am not going into detail of that. So this is basically the fundamental structure of any amino acid that we encounter. So when dispersed in a solvent, and there are certain polar groups present here, right? COOH and NH2, these are the polar groups. So immediately they get ionized, and they create a dipolar behavior in the amino acid. So one side of the molecule becomes partially positive, another side becomes partially negative. This is also a particular characteristics that is always found in all amino acids. In the amino acid, there are these two functional groups, NH2 and COOH, are fixed, and these groups, if you see to it, then NH2 group is a basic group, right? You know that NH2 has a fundamental basic property associated with. On the other hand, COOH is the carboxylic group, and it represents acidic behavior. So physiologically, when they exist in this form, in this dipolar form, then they are commonly called Zwitter ions. This is the figure that represent these ions, right? So popularly we represent this by this way, NH2 plus CHR. So remember, this is CHR, COO minus. This is the Zwitter ions structure. So basically this is what amino acids are. As soon as you change one by one the R, this R group, this functional group changes, you get different amino acids. So in general, there are almost 300 amino acids present in the nature. Of these, 20 amino acids are present in all forms of life. The same amino acids you can find in plants, in animals, in bacteria, in every living being you can find these 20 amino acids. Other amino acids are also present, but in a much smaller amount, and they are not that much universal, so to say. So this universal nature of genetic code that needs to be read is basically the underlying truth of having these 20 basic amino acids. They are called standard amino acids. Other than these 20 amino acids present in the present, there are numerous other amino acids which are non-standard amino acids, right? So in terms of these 20 amino acids, let us try to understand these 20 amino acids better. So of these amino acids, they have different kind of properties, and based on that we will also talk about their structure a little bit. So of these amino acids, let us first talk about that who are called essential amino acids. Essential amino acids is a group of amino acids. They cannot be synthesized inside human body. So this is particularly for human. In particularly human, the amino acids like histidine, arginine, valine, this list, if you go through this list, these are the amino acids that are not synthesized inside the human body. However, histidine and arginine can be synthesized in adult body sometimes, but not in children, and they can also be considered as a semi-essential amino acids or so. So all the rest of the amino acids, rest of the 20 amino acids, are non-essential or dispensable amino acids because they can be formed inside the body. So we separate them in three groups: essential, conditionally essential, and non-essential or dispensable amino acids. Now let us talk about these amino acids in more of a structural sense. So if you look into the structural sense of structure of these amino acids, you will find that some of them are hydrophobic in nature and some of them are hydrophilic in nature. So for example, these are example of certain hydrophobic amino acids. So hydrophobic amino acids will be glycine, alanine, valine. They are all hydrophobic amino acids. That means that they are afraid of water. If you they don't like the water to be in vicinity of these molecules. On the other hand, some other amino acids can also be hydrophilic amino acids like this. So for serine, asparagine, these are amino acids which are hydrophilic amino acids. So in hydrophilic amino acids, you will always find a functional group where you have CH2OH, CH2SH. This kind of groups will be present here. On the other hand, in case of hydrophobic amino acids, you will see H or CH3 or similar groups present. So thus you distinguish between these two amino acids, these two kinds of amino acids. So amino acids are held together one by one, they bind together one by one like a long chain, forming a long chain. And that is what is called a polypeptide, which is a very initial form of a protein. So the bond that holds them together is called a peptide bond. Peptide bonds are when you have two amino acids side by side, and as you remember, always they will have one COOH group and one NH2 group. So when these two groups combine, a peptide bond forms. So let us focus on this part. What happens is this OH got coupled with one H from here and a water molecule was exited, and you have a CO bond NH and then the second amino acid. So this is basically the way one by one these amino acids get stitched, forming a long polymer chain. The peptide bond is a rigid and planar bond. Remember that it's a planar bond, and it generally exists in a form when long chains, in every long chain you can have these peptide bonds present in when you are dealing with a polypeptide or amino acid chain. This particular kind of reaction where this happens is called a polycondensation reaction, when the carboxyl group and the amino acid of one amino group and carboxyl group, they combine and they lose one water molecule. This is called a polycondensation reaction. So molecules where you have one by one stitched like this 100 amino acids, around 100, you call them polypeptides. Polypeptides are when the length of the chain is around 100. And when you have more than 100 of this number, then it is going to become a protein later on. So X-ray structure analysis reveals that this peptide bond which attaches these two amino acids has a specific planar structure, and the planar structure is where all the four atoms in the peptide bond lie in one plane. So all of them, all these four atoms lie in one plane. Okay, so remember that this is a planar structure of the bond. So when you have this kind of a bond forming one by one, then obviously we are moving towards a long polymer chain, and these long polymer chains are nothing but proteins. So whenever we say protein, that basically means that more than 50 amino acids are combined. This is still in a polypeptide chain, but it can become a protein if it folds properly and gets a 3D structure. So protein's biological activity is not manifested by what are the amino acid, only not that part, but also the folding is very, very important, and the 3D structure, the 3D conformation that the protein has is very, very important. So there are different conformational states of a protein, and we can divide them in a 4 hierarchical levels. The first one is called primary structure, second one is called secondary, tertiary, quaternary structure. So let us one by one talk about how these structures are formed. So the primary structure largely says that how the amino acids are combined. So that is mostly amino acid chain. Here the sequence of the residues, amino acid residues, is most important, and we write them using the alphabetical code for each amino acid, right? So that we have learned in the while learning about genetic code. So as we know that in the genes present, there are DNA to mRNA, then tRNA plays their role, and then the amino acid sequence is made. That is called a primary structure. So this is the primary structure where you have the amino acid chain, where ribosome have read out the genetic code and one by one combined the amino acids in a particular chain. Remember that in this form, this primary protein structure is not biologically active. They cannot perform any task that is assigned to you. This is just the polymer which have been prepared. The secondary structure is one of the folding strategies, right? So in the secondary structure, what happens is this can be a biological active state of the protein molecule. So in here, two structures are seen. One is called alpha helix and one is called beta sheet. So let us talk about them separately. Alpha helix is basically a tightly packed coil-like structure around the central axis when the amino acid chain extends outwards and it coils around a particular axis. This structure is maintained by formation of large number of hydrogen bonds. Both kind of helixes can be made. Helix like this, that means helix where right-handed helix or left-handed helix. Both kind of helix are made. Right-handed helix are more stable, and there is of the order of nanometers, 0.1 nanometers or so space between each amino acid when this particular structure is made. On the other hand, beta sheets, so this is alpha helix, right? Alpha helix, this is a helical structure. You can see it is rotated around a particular axis. Beta sheet is something where you have two peptide chains which are side by side, either parallel or anti-parallel in structure, and then hydrogen bonds start forming between them. Two polypeptide chains side by side, two or more polypeptide chains side by side, and then hydrogen bonds start forming between them. So this structure can be formed by multiple chains, or maybe a single chain which has folded upon itself. So a single chain might come back and fold upon itself, and then you can have this typical beta sheet like structure. So let me give you an example of how beta sheets could be formed. Here you can see two amino acid chains, two polypeptide chains. This could be the same polypeptide chain which has folded back upon itself, and here you see how the hydrogen bonds are forming for the same reason, for the polarity of the amino acids, the polar behavior of the amino acids. This can be formed. Okay, so this is a biologically active, this could be a biologically active form. Now comes the tertiary structure. Tertiary structure of the protein is a much more compact arrangement where the hydrophobic sides of the chain, so we just talked about that some amino acid residues are hydrophobic and some are hydrophilic. So hydrophobic side of the chains face towards the core, so they rotate such that inside this area where no water would be there, near the core the hydrophobic part of the chain goes. On the other hand, hydrophilic parts are directly towards the periphery, outside. So other than hydrogen bonds, several different kind of electrostatic interactions can also happen, while making this structure, right? So around 50 to 350 at least amino acids are required to make it fold like this, and this is a very much biologically active structure where they can perform specific tasks. The quaternary structure is complicated than this as well. Quaternary structure is when you have two or more such structures like tertiary structures, they combine and join into a single protein where you have multiple subunits. So suppose you have two such folded structure where the hydrophobic parts are inside, hydrophilic parts are outside, several hydrogen bonds have formed, and then two such structures are again combined together to create a new protein, right? A single protein. So this is a single protein that has two different subunits. So a common way to describe this quaternary structure is through naming these particular proteins present. For example, suppose a molecule is designated as alpha, beta, gamma. The example that I have written here, suppose a molecule is written as alpha, beta, gamma, it indicates that there are three distinct peptide chains, right? One of them I am calling alpha chain, one of them I am calling beta chain, one of them I am calling gamma chain. Three different parts, three different chains who are different peptide chains. They are combined together as subunits to create one molecule, one protein molecule. Similarly, if I say say that we have a protein which has a alpha 1, beta 2, gamma 2 structure, then what does it mean? That means alpha 1 means only a single alpha chain, that means one kind of chain which I am calling alpha, that is only one chain. Two beta chains, that's the second kind of chain which I am calling beta, and a third chain which I am calling gamma, two components of that. So these 5 subunits, one alpha, 2 betas and 2 gammas, they are combining together to make one large protein molecule. These kind of structure are most common in regulating cellular function and metabolism. Hemoglobin is a common example of this structure, and we started our lecture today with the structure of hemoglobin, and now you will understand it's a tetramer consisting 2 dimers that bind to oxygen, and it is oxygen binding protein in the red blood cells. We will all know that, right? So this has a quaternary structure. In this structure, you have 4 peptide subunits, the blue ones and the red ones. So it's a alpha 2 beta 2 structure. 2 polypeptides which have similar structure, the red ones, and 2 polypeptides who are exactly identical are called beta molecules. So thus they are all coupled together with 4 subunits, and this is a functional protein that is performing and doing its task for binding oxygen inside human body. So this is more or less about protein structures today. We will continue in the next class.